Hi, my name is Malvika Sudhakar and I'm from IIT Madras. And today I'll be presenting my poster BinOpt, an algorithm to optimally assign feature importance to classes. A large number of biological problems can be formulated as classification problems. For example, one might be interested in classifying uninfected cells from infected cells, or say disease samples from normal, or say the different stages of cancer. So these are our classes. The data used for classification can be from mutation data or gene expression data, in which case the genes are our features. So typically in biology, we have a large number of features, and hence dimensionality reduction is used to identify a minimal subset of features that can distinguish between the classes. But what do we mean by feature importance to classes? Let us understand with the help of a toy example. So here the classes are the different colored bins and the features are the different colored balls. The balls define which colored bin it is. So when we have a subset of these features, for example, the yellow balls, we cannot distinguish between the two bins. But when we have other subsets, for example, the yellow and the blue balls, we can now differentiate between the two bins. Similarly, when we have the red and blue balls. So there exist multiple equivalent feature sets that can distinguish between the bins. This is especially relevant in biology where the features are correlated and you have multiple equivalent subsets that give equal classification power. Now, the question we asked is, can we use these feature sets, which are equivalent, to find the feature importance in bins? So the equivalent feature sets are represented as matrix S, and the feature importance to classes is represented by matrix X. So let us look at the formulation for BinOpt. Starting with the assumptions for matrix X. The first assumption is that each feature belongs to at least one bin. The second assumption states that the bin cannot be empty. And the third assumption states that you should have a feature that can differentiate between a pair of bins. With this, we come up to the assumptions for the subset, and we say the subsets are created by the first assumption being the subsets are created from one feature from each bin. And the second assumption states that you should have a feature that can distinguish between the two bins. Now, with this, we formulate our optimization problem where the first three constraints are basically the assumptions for the matrix, and the next two constraints are the assumptions for the subsets. And the last constraint is the sparsity constraint that controls the sparsity of the predicted matrix X. And this is especially important because the objective tries to minimize the number of ones in matrix X. So what this means is we try to reduce the number of bins a feature can belong to. So we then tested out the algorithm by simulating various matrices and created subsets from these random matrices, which were given as input to the algorithm along with the sparsity. And then we compared the predicted matrix with the random matrix. And what we find is that when we look at the sizes of the matrices, it doesn't seem to have a significant effect on the prediction of the matrix, unless the number of bins are 2. Similarly, when we look at the percentage of subsets or the fraction of subsets given as input to the algorithms, where 100 denotes all possible subsets given as input and 20 represents 20% of the subsets given as input to the algorithm, again, we see that it doesn't seem to have a significant effect on the prediction. And this is especially important because in real world problems, we might not always have all possible subsets, but we might only have a fraction of subsets. And what this shows is that we can still predict the matrix given a fraction of subsets. Now, the same is not true for sparsity. We find that the algorithm performs well only when the sparsity is very high or very low. And then we looked at the importance of the sparsity constraint. And as you can see, without the sparsity constraint, the prediction of the matrix reduces as the matrices get denser. Now, this assumes that the sparsity of the matrix is always known, which might not be the case. But what we might know is the range in which the sparsity falls in. So we developed 6 methods for estimating the sparsity of the matrix, and we found the dense method works best on dense matrices and the normalized method works well on sparse matrices, which can be further seen by this plot of true sparsity against the estimated sparsity. We then employed BinOpt on a real-world biological problem to find out the gene contribution to cancer types. The data out here is 21 cancer types for various genes, and the genes are the features and the cancer types are our bins. We generated subsets from the mutated samples, and we ran pairwise BinOpt for all pairs of cancer types and took the consensus for the results. What we observe is that the genes that we identified overlap with the genes in the cancer gene census, and they also were enriched for cancer pathways. And there were some genes which are not found in the CGC list, which might be potential driver genes. To summarize, we show that BinOpt can identify feature contribution to various bins given equivalent subsets of features. We further showed the prediction capability of the algorithm for various input and matrix parameters. We then discussed about 2 methods for estimating the sparsity of the matrix, normalized and dense for sparse and dense matrices respectively. And lastly, we employed BinOpt to find the contribution of genes to various cancer types. Thank you for your time.